So if you were going to perform these steps in a laboratory, you would take the DNA that you wanted to make copies of, put some of that into your tube. Then you would need polymerase, put that into your tube. And then you would, of course, need your extra nucleotides and primers. Put that into your tube, and now that you have your tube full of all this good stuff, you would need to take that and put it at 95 degrees Celsius for a minute, and then you would move it to 55 degrees Celsius for a minute, and then you would move it to 75 degrees Celsius so that you could do all three phases of PCR, and then it would go back to 95 degrees Celsius, and you can imagine yourself standing there holding this little tube and putting it into these 3 water baths for about 2 hours. It would be tedious, but for getting 1 billion copies of your DNA, it's probably worthwhile. Luckily for us, the process is now pretty much automated. So you would take your DNA, put that in your tube. You would take your polymerase, put that in the tube. You would take your primers and nucleotides, put those in the tube, fill it up. You would then put it into this machine called a thermal cycler, which is basically a really well-calibrated heating machine that'll put it to the 95 degrees and 55 degrees and 75 degrees in a cycle, and you can basically walk away, go out, get some lunch with your friends, come back, and 2 hours later, come back, and you have 1 billion copies of your DNA. So this is incredibly convenient in a genetics laboratory. It's a good way to make a whole bunch of copies of your DNA. The only downside to this, and the reason we won't be doing it at the high school level, is if we were to look at the price tag on this machine, you're looking at minimum $5,000, probably for a good one $20,000. So for our purposes in lab, it's easier to buy some DNA that's already been amplified rather than have this machine to amplify our own samples.